Ever wondered how your body turns sugar into energy? Well, today we're diving into one of the most important biochemical processes, glycolysis. Whether you're powering through a workout or just keeping your body going throughout the day, glycolysis is working hard to give you that quick energy boost. But how does it all happen? Stick with me, and let's break it down step by step, from glucose to pyruvate. Let's get started. The story of glucose starts with the food we eat. Foods rich in carbohydrates, like bread, pasta, and fruit, are broken down by enzymes in our digestive system. This process begins in the mouth and continues through the stomach into the small intestine. In the small intestine, the resulting glucose molecules are absorbed through the intestinal walls and enter nearby blood vessels. From there, glucose is carried by the bloodstream to every cell in the body, where it can be used for energy, starting with glycolysis. Before glycolysis can begin, glucose needs to enter the cell. After digestion and absorption, glucose travels in the blood and reaches various tissues. But it can't just slip through the cell membrane. It's too large and the membrane is selective. Instead, glucose enters through special proteins in the membrane called GLUT transporters. These act like doors or gates that let glucose in, but only when it's moving from a high concentration outside the cell to a lower concentration inside. This process is called facilitated diffusion, and it doesn't require any energy. In muscle and fat cells, insulin helps by increasing the number of GLUT4 transporters, making it easier for glucose to enter. Additionally, some cells, like liver and muscle, don't always need external glucose. They can break down stored glycogen to get glucose internally, especially when you're fasting or exercising. Now that glucose is inside the cell, glycolysis can begin. The first step is called phosphorylation. We add a phosphate group to glucose, forming glucose 6-phosphate, or G6P. This is catalyzed by the enzyme hexokinase in most cells, or glucokinase in liver cells. One molecule of ATP is used to donate the phosphate. This step is important because G6P can't leave the cell. The phosphate group makes it charged, so it's trapped and ready for further breakdown. In step 2 of glycolysis, the 6-carbon glucose 6-phosphate is rearranged into another 6-carbon sugar, fructose 6-phosphate. This is done by the enzyme Phosphoglucose Isomerase. It's a reversible reaction and doesn't require energy. The change prepares the molecule for the next key step, another phosphorylation that locks it into glycolysis even further. This step is often called the commitment step of glycolysis, because once this happens, there's no turning back. The enzyme Phosphofructokinase 1, or PFK1, uses another ATP to add a second phosphate to fructose 6-phosphate, creating fructose 1,6-biphosphate. This step is irreversible and heavily regulated. The cell decides whether to move forward based on how much ATP or AMP is present. When ATP is high, PFK1 slows down. When the cell is low on energy, PFK1 speeds up glycolysis. Now that fructose 1,6-bisphosphate has been fully activated, it's ready to be split in half. The enzyme aldolase breaks the 6-carbon molecule into 2 3-carbon sugars. One is glyceraldehyde 3-phosphate, G3P, and the other is dihydroxyacetone phosphate, DHAP. Only G3P will move directly into the next steps of glycolysis, but don't worry, the DHAP will be converted to G3P in the next step. In the previous step, we got 2 different 3-carbon molecules, G3P, which continues in glycolysis, and DHAP, which can't. So, in step 5, the enzyme Triose phosphate isomerase converts DHAP into G3P, making 2 identical G3P molecules. This reaction is reversible and very fast. In fact, it's one of the most efficient enzymes in the body. Now both 3-carbon molecules can continue through the rest of glycolysis. Now that we have 2 G3P molecules, glycolysis enters its energy payoff phase. In step 6, each G3P is oxidized by the enzyme glyceraldehyde 3-phosphate dehydrogenase, or GAPDH. During this oxidation, NAD is reduced to NADH, a molecule that can later be used to make ATP in the mitochondria. A high-energy phosphate is added to G3P to form 1,3-biphosphoglycerate 1,3-BPG, which has a lot of stored energy. Since we started with 2 G3P molecules, this step produces 2 NADH molecules. This is where glycolysis starts to pay off. In step 7, the high-energy molecule 1,3-biphosphoglycerate donates a phosphate group to ADP, forming ATP. This is called substrate-level phosphorylation. ATP is made directly from a molecule in the pathway, not through the mitochondria. The enzyme Phosphoglycerate kinase, PGK, carries out this reaction. Since we have 2 1,3-BPG molecules, this step produces 2 ATP, recovering the 2 ATP that were spent earlier in glycolysis. The product of this reaction is 3-phosphoglycerate 3-PG. In step 8, we don't add or remove anything, we just rearrange the molecule. The enzyme phosphoglycerate mutase shifts the phosphate group on 3-phosphoglycerate from the third carbon to the second carbon, forming 2-phosphoglycerate 2-PG. This setup is important because it positions the phosphate in just the right spot for the next step, where energy will be released by removing water. In step 9, the enzyme Enolase removes a water molecule from 2-phosphoglycerate, forming Phosphoenolpyruvate, or PEP. This dehydration reaction might seem small, but it creates one of the highest energy molecules in all of metabolism. PEP is perfectly set up for the final step, where it will transfer its phosphate group to make ATP. This is the final step of glycolysis, and it's another big payoff moment. The high-energy molecule PEP, Phosphoenolpyruvate, donates its phosphate group to ADP, forming ATP. This is carried out by the enzyme Pyruvate kinase, which is highly regulated. The product is pyruvate, a key molecule that can go on to the citric acid cycle in the presence of oxygen or fermentation without oxygen. Since there are 2 PEP molecules per glucose, this step generates 2 ATP, giving us a net gain of energy. At the end of glycolysis, we're left with 2 pyruvate molecules. But what happens next depends on whether there's oxygen. If oxygen is available, pyruvate enters the mitochondria, gets converted into acetyl-CoA, and joins the TCA cycle, leading to more ATP through oxidative phosphorylation. If oxygen is lacking, like during intense exercise, pyruvate is converted into lactate by the enzyme lactate dehydrogenase. This process is known as anaerobic glycolysis, and it helps regenerate NAD, so glycolysis can continue and keep making some ATP. So, pyruvate is a fork in the road. Aerobic, more ATP, or anaerobic, quick energy with lactate. Let's wrap it all up. Glycolysis starts with glucose and ends with 2 pyruvate molecules. Along the way, it makes a net gain of 2 ATP and 2 NADH, which can be used for more energy later. It happens in the cytoplasm, and importantly, it doesn't require oxygen. That's why all cells, even without mitochondria, can use glycolysis. Pyruvate then either enters the TCA cycle if oxygen is present or gets turned into lactate if it's not. Glycolysis is fast, efficient, and essential, the universal first step in getting energy from glucose. And that's a wrap on our deep dive into glycolysis. If you found this video helpful, make sure to hit that like button and subscribe so you don't miss out on more science breakdowns. Until next time, stay curious and keep learning.